The European Journal of Neuroscience is a society journal for the Federation of European Neuroscience Societies. It's your journal. It's the place where many people in the world community come to publish good, solid, well-conducted, rigorous science.
Society-owned journals have a critical role to play in publishing. One part being because societies represent the collective view of the scientific community, and also the manner in which they view publishing at that point in time. And I think that's very important because publishing is a changing situation.
Every second year for the FENS Neuroscience meeting, we have a Young Investigator Award and a Senior Investigator Award. And it's a way for the European Journal of Neuroscience to partner with its society, Federation of European Neuroscience Societies, FENS, to recognize outstanding work by an established figure in the field and by an up-and-coming, super exciting youngster who's just blazing a trail.
These awards, they are actually sponsored by Wiley, the publisher of EJN, and given by a committee from FENS. And these awards are here to recognize outstanding contributions in neuroscience.
Recognition or highlighting of specific work done by an individual opens up that work to a larger and broader audience that may or may not otherwise get to see it.
Is the recognition of your own community to the work you do. So it's a specific and very special recognition. Is your colleagues telling you that you are doing a great job.
Our senior award winner this year is Seth Grant. And in some ways, Seth really embodies every aspect of what you want to see in the senior awardee for the EJN FENS prize. He's a senior researcher who's made fundamental contributions to our understanding of the mechanisms of behavior and how those mechanisms are involved in brain disease. He's based in Edinburgh, and FENS this year, of course, comes to you virtually from Glasgow. So it was a fantastic year to have somebody based at a major Scottish university while FENS was being hosted by the Scots. He represents the incredible international breadth of the FENS community. He hails and did his original work in Australia. What I love is that the title of his talk this year is very exciting because I think we're going to be taken on a really wide-scoping journey of a great body of work, the synaptome, an epic journey in time and space.
It was 20 years ago at the Federation of European Neuroscience meeting in Brighton that I had the privilege of giving a lecture where I reported our recent discovery of a very large number of proteins that were found inside synapses in the mammalian brain. And this really was a turning point because we suddenly realized that synapses are not just simple connectors with a few molecules, but in fact are highly complex, sophisticated computational devices. It also was a turning point because the discovery of these proteins opened up the door on understanding many brain diseases. At the time, we found there were 3 of those proteins, which were known to cause genetic neurological conditions, but were not known to be acting in the synapse. So we now could suggest that synapses and their proteins were going to be the target of many genetic disorders. And we studied that a lot over the next decade. And in 2011, we showed that by studying the human synapses, that there were several hundred genes responsible for more than 130 different brain diseases, which affect the synapse proteins as well. So this has really been a major sort of transition in the way we think about synapses and their role in human behavioral disorders. This area of work, which is through the study of proteins, has now taken a very interesting new turn, which is to ask the question, which synapses contain which proteins? Now, this is, you might think, a rather uninteresting problem because we know about some synapses called excitatory and inhibitory synapses. But actually, when you use molecular methods, which allow you to look into individual synapses, but on a very large scale, we found that there is a vast and potentially unlimited diversity of synapse types in the brain. And by using methods which allow us to map them across the entire brain, we discover that this diversity is organized into something called the synaptome architecture. I should just mention, we're used to the word gene and genome, being all the genes, proteins, proteome, all the proteins. And you can think of synapse and synaptome being the full complement of synapses. And so these methods that I'm referring to are now called synaptomic methods or synaptome methods, which allow this sort of large-scale, systematic and comprehensive analyses of individual synapses and their molecular compositions across the brain. Having given talks at FENS over many years, it's a very special thing to be recognized by your peers for the body of work that one has accumulated over so many years. So yeah, that means a great deal to me personally, but also I consider it a very important thing for all those people who have had the privilege to work with, because it's not just me who does this work. I've had the privilege of wonderful postdocs and graduate students. Some of them have been extremely innovative and have done very important contributions to all of this work. And so I think this is really an award to them as well.
This year's recipient of our Young Investigator Award is Laurence Hunt.
I think one of the major questions we all try to address in neuroscience at various levels is how can you go from the molecular and circuit level neuroscience to how this results in global activity of brain and how this controls behavior. So the study of the process of decision making is one of the instances in which we are starting to have, we as a field, of course, are starting to have a good understanding of what are the mechanisms at all levels. And so I think the work of Laurence Hunt has been a major step forward in making this link. And therefore, I'm sure it will be of interest not only to the specialists of the prefrontal cortex, but to a much wider audience.
It's a huge honor, obviously, to be recognized by FENS in this way. I think it's particularly exciting for me because FENS was one of the first conferences that I attended when I was a first-year graduate student, back when it was in Geneva in 2008 or so. And I remember being excited at the time, not just because people would come up to my poster and would be interested in my work, but also because it gave me a chance to present to people across the whole broad spectrum of different areas of neuroscience. And so I think receiving this award is similarly very exciting in that it gives me a chance to share my work over the past 4 or 5 years with people from across the spectrum of neuroscience. So my research at the moment kind of has 2 strands. The first is going to build upon some of the recent findings that I'll be presenting in the lecture. And this is to study the role of different regions of prefrontal cortex during reward-based decision-making. So the aim of this is to link some of the representations that we found in different parts of monkey prefrontal cortex to equivalent representations in human imaging data. So the way that we're doing that is using a technique called representational similarity analysis. My current work there is also to try to develop behavioral tasks that kind of move towards studying decision-making in slightly more naturalistic and temporally extended settings. So a lot of the tasks that I and others have used in the past have tended to use really simple decisions where everything is presented to the subject simultaneously when they're asked to make the decision. And decision-making in the real world doesn't look like that. You know, we shift our attention around as we make choices between different options. We have to make long-term plans. We have to seek out information about different choices. So I think that there's a lot of work still to be done in kind of building formal frameworks that allow us to study more naturalistic decision-making and how different regions of the brain support that. So that's one line of research which I'm currently pursuing. The second area is a much more practical area of research, which is to try to capitalize upon some of the really kind of well-established paradigms in cognitive neuroscience, the ones which have been developed over the past 10 or 15 years, and try to refine them in such a way that they might become more useful tools in the clinic, either in psychiatry or in neurology. So I think that the challenge there is really a practical challenge of how do we refine those paradigms so that we could try and get a meaningful signal from quite noisy human imaging data, and we try and do it at the level of individual subjects rather than at the level of groups of participants. I think that it's a real shame that we're not able to meet in person in Glasgow this year because both with Brexit and with the coronavirus, I think that there's a risk that we lose some of the key opportunities that are needed for scientific collaboration and networking across Europe. But on the other hand, I think that by making the most of kind of these newfound skills that we're developing in communicating and conferencing virtually, this might actually lead to strengthened international networking in the long run. So I'm really glad that the virtual forum is going ahead and that the Young Investigator Award gives me a chance to be a part of